So now is the turn of Birol Aydin. Mr. Birol is a clinical senior embryologist and scientific director at Ovogene Bank with over 15 years of experience in embryology and andrology across 20 countries. He has set up 10 IVF labs, has conducted over 50,000 ICSI embryo transfers, 20,000 PGT cycles, and 15 egg donation cycles, as well as 122 spindle nuclear transfer cycles, with achieving 22 pregnancies and 12 live births. He has published and participated in more than 15 papers and contributed to advancements in genetically certified oocytes, AI for oocyte morphology, and time-lapse vitrification imaging. He's editor of Elsevier's cryo guidelines, and today we'll share his results with the ultrafast vitrification and ultrafast warming protocol, highlighting not only the survival and clinical outcomes, but also the transcriptomic analysis. So please, Birol.
Good afternoon, everyone, and thanks to Kitasato for our collaboration, support, and invitation. So as we know that ultra-rapid vitrification and warming now is the trend topic in IVF area, so because it's making our job more easier, more efficient. But of course, when we get some technologies or techniques to our laboratory, the main concern if those technologies are really safe for our patients. So we are here now more focused on the safety side and efficacy side of ultra-rapid vitrification and warming. So first of all, we have to understand that to find out the safety of the one lab techniques, there are two ways to do it. The first way, as a traditional way we many times are doing, this is a beyond of the research, of course, we just adopt these technologies to our laboratory and to use and to find out the clinical result for the patient. If that clinical results are good, as we are thinking, okay, this technology is fine and we can adopt to our laboratory. But of course, as a scientifically and ethically situation a little bit different, because this is long term of the life of the human. So we are part of creation of the human, and we have to understand that this really should be safe and efficacy for the patient. So the second way, of course, to check the transcriptomics. So the transcriptomics may detect at gene expression level, which is very difficult. In whole cell biology now, there are only one research for the mouse oocyte stage for the transcriptomic, which is limited only with 2 samples. So our study is the first large-scale testing for the single-cell gene expression on the RNA-based. We already tested around 48 samples, which I will explain that how study was working. So as of course oocyte, there are different stages, immature stage, mature stage, so that different gene expression we are getting from the RNA level. So we are here the focus mostly for the maturation stage, the fertilization stage, and also embryo development stage for the transcriptomics. As on the embryo stage, of course, that we have also different level of the embryo development. As a morphokinetically, we were testing for the zygote stage, day 3 stage, and also day 5 and day 6 embryonic development stage to identify the critical gene mutations, which is mostly effective for the molecular stress level, which is increasing because of the techniques or technologies. As mainly that this hypothesis is very important when we accept one technology or technique to our lab, we have to evaluate very well. So firstly, we created as hypothesis, okay, hypothesis is saying ultra-rapid vitrification and warming can increase the efficacy and efficiency. But from second side, that we have to make the immediately animal study. After animal study, so we have to understand that the structure of the oocyte on the animal and human, there are differences. For the zona permeability, for the zona birefringis and the spindle configuration, it shows differences. So that means if you are receiving with the mouse animal study that a high survival rate, that doesn't mean with human oocyte you will get the same, because there are different parameters and the components which is effective here for the post-ICSI timing, for the morphokinetic development for the lab condition for the animal model and human model are totally different. So where idea came, we want to check transcriptomics because it's very difficult to create an RNA library. The idea came from the publication which published by the Chinese group, and they declarate that after the frozen embryo transfer, they found significantly higher novel mutation for the frozen embryo transfer cycles, comparing natural conception and regular development. So that's why we thought that if we check the transcriptomic level, that will not give answer only for ultra-rapid, but it will give answer and will open way for the many new techniques and technologies which we are adopting to IVF laboratories. As the main challenging for the cryopreservation is basically for oocyte stage is the low survival rate. Of course, ice crystal formation, we mostly manage that with the conventional vitrification, but still that we couldn't achieve high survival rate on the oocyte stage. So that osmotic stress risk and the cytotoxicity risk, it was still considerable, and we were searching answer to those questions as well. So between ultra-rapid vitrification and standard conventional vitrification, basically the conception of the media is the same. So we have no huge difference. The aim here to use just the different steps of micromanipulation. So to decrease the micromanipulation, to decrease the timing of cryoprotectant exposure, and that way we will keep more stable to molecular stress, and we will increase the gene expression rate and to keep stable and protected. The same thing for ultra-rapid warming. So for ultra-rapid warming, especially for the donor egg banking, it's very crucial because we are freezing most of us as a frozen cryo egg, and we have to thaw them with a small batch of eggs in the safety and high efficiency. Of course, here it is very important to increase survival rate that will affect directly cumulative blastocyst rate as well. So the same thing, either 1.2 molar trehalose, either 1.0 molar sucrose. So the one-step thawing solution that will solve exchanging time of the 60 seconds between cryoprotectant and the water exchange as a safety. So the traditional technique which we were keeping like 1 minute on the TS solution, and then we were moving that half molarity of the DS solution, we were increasing two risks. The first risk, of course, because the molarity exchange, the zona permeability get lower, zona birefringence get lower, and the most of the eggs were degenerating on the dilution solution stage as DS stage. So ultra-rapid warming to avoid the help to minimize the micromanipulation, especially which is coming through the lab condition and skills of the embryologist. From this perspective, it brings big advantage for the high survival and blastocyst efficacy. As also study shows between cryopreservation, different timing of the spindle configuration, actually there is no huge difference between the conventional vitrification and ultra-rapid vitrification for the spindle formation. However, there is a difference for the molecular level on the endoplasmic reticulum function and also the mitochondrial function. As the same study, we're focusing of the fresh and ultra-rapid and frozen cycle with ultra-rapid to different cell stage as immature oocyte as GV, M1 oocyte and M2 oocyte. So the study aimed to show with the ultra-rapid vitrification, we are independent from the oocyte quality. That means any kind of the oocyte, the different maturity stage still can survive. But of course, efficacy of the blastocyst rate or cleavage arrest rate depending of the potential of the eggs. As our study, we're analyzing mostly around 1,700 oocytes, the sibling oocytes between conventional and ultra-rapid with the 6 pilot clinic, which has a different experience of the embryologist, different lab condition, and they were focused on to make their own culture media. Each of them are using different. So we aim to get ultra-rapid vitrification is independent also from the embryologist skill and the lab condition. So here the most important data what we get, the comparing conventional rate for such many oocytes, 57.34% of the used blastocyst rate, comparing conventional warming and vitrification, that is pretty high. Of course, on this stage, euploid blastocyst rate also increasing. As our transcriptomic study, the objective was to evaluate the metabolic and secretome phase of the embryo development and also ultra-rapid vitrification, how affect in the mid-infrared spectrophotometry, also RNA sequencing. So what we did, we used around 1,214 sibling donor oocytes. We used them the two groups. Half of them freeze by ultra-rapid vitrification and half of them freeze by conventional vitrification. And we thawed those oocytes same as ultra-rapid and conventional warming. And we made ESHRE embryo culture. And on next stage, we were checking for the secretome profiling as lipid concentration, protein concentration and structure, nucleic acid concentration and structure. We were following them till day 6 embryo development, and the IVF outcomes as a survival rate, cleavage rate, blastocyst rate, and the euploid rate. And the final steps was RNA analysis. For metabolic stage, we use spectrophotometry. As on the spectrophotometry result, as you see between conventional and ultra-rapid vitrification and warming, based off DNA concentration, saturated lipid concentration, protein structure, and the nucleic acid concentration and structure, graphics show similarity. That means at the metabolic level, between ultra-rapid vitrification and conventional vitrification, there is no any harmful difference for the oocyte stage. So for the transcriptomic analysis, as we have the pilot study, we used 24 trophectoderm biopsy sample and 24 oocytes. So each group we separated 8 trophectoderm biopsy sample and 8 oocyte sample for fresh ultra-rapid vitrification and the conventional vitrification. Our aim checked to their transcriptomic level. So we use the heat map technology for the detecting of the single gene sequencing for RNA level. We use poly-A selection for the transcriptomic panels for the gene expression. We avoid and disclose from the study for the ribosomal proteins. As the quality control, we had very high reading level, so the old samples had a result. Only one sample from the fresh cycle, we couldn't get cDNA, so those samples still need evaluation. So as a morphokinetic parameter, we were checking the post-ESHRE degeneration, which is important, cryosurvival rate, and also we were checking high-quality day 5 blastulation rate. So which is very important here, post-ESHRE degeneration rate in the conventional group was 5.7, but ultra-rapid group 0.8, which is very, very low. And another limitation which we are getting from the cryo oocytes is high-quality day 5 blastulation. So it was conventional group 29.9, which I am talking only AA, AB, and BA quality, and the ultra-rapid 46.5, almost double, which is very high. As heat map analyzes, as a principally how system is working, so we are checking the different, many of the gene expression, of course, many of the gene mutation. So as you see in the left side, those are for oocyte, and the down, which you see, those are for the trophectoderm biopsy. So the localization of the oocytes for the fresh, frozen, and ultra-rapid is the same place, but when you see the demographically show difference for the trophectoderm biopsy, so that make us big concern as trophectoderm biopsy or blastocyst freezing for ultra-rapid vitrification and warming still may not be safe for perspective of the transcriptomic and novo mutation. So for oocyte, we get high efficiency, but for blastocyst, we have concern. As a conclusion, so we can say ultra-rapid vitrification and warming can keep cell viability and cell structure in high level, and it can keep also the mitochondrial function and organelle function in the maximal stage. Also, it's shortening time, and limited micromanipulation can increase the efficacy and efficiency of the embryo development and the survival rate. And the one-step rate rotation protocol also allows to make the faster lab job, so to decrease the post-ESHRE degeneration and increase blastocyst efficiency. Thank you very much.
Thank you very much, Birol, for your amazing presentation. Congratulations for your